Assalamualaikum warahmatullahi wabarakatuh, class. Now we are on a new chapter for chapter special senses. So this closely related to the neurons nervous system that we have learned on previous chapter. So mostly for the special senses are directly related to the sensory neuron. So there are types of special senses, whether it is from the chemical, it is from gustation and olfaction, or from the sight that we can see, what we call as eye and vision. And we do have special senses of ear and hearing. So at the end of this lesson, you should be able to describe the sense of taste, describe the sense of smell, what are the structure, what are the function of eyes, photoreception, what is it, visual accommodation, explain the homeostatic imbalance, functions of the ear, mechanism of hearing, and balancing. So we go for the first part of chemical senses. We do have gustation, what we can taste by the tongue, and the olfaction, what we can smell with the nose, and both dependence on chemoreceptor. Why we call it at chemo? We do have the chemicals. Where are the chemicals? The chemicals, for examples, the taste receptor, we do have the food chemicals. So these chemicals can dissolve in saliva. For second one, for the smell, we can have the smell of airborne chemicals. So they can be dissolved in fluid coating nasal membrane. So most of them will be dissolved in this aqueous solution, whether it is in saliva or without or within the nasal membrane of the liquid or fluid. So the olfaction, what we can smell, we do have the olfactory receptor cells. Where is it? Okay, so we do have the nose, means all the hole there. So from the hole, we do have this nasal cavity. The nasal cavity, at the upper part, we do have olfactory receptor cells. The nasal cavity there, if we enlarge the illustration, you can see it's starting from the mucous layer. So from the mucous layer, it has cilia of receptor cells. So the receptor cells are like a hair-like structure, cilia, okay, when we call it, and it is transferring or transmitting the impulse to the receptor cells. From there, it's going to the, lastly, to the olfactory bulb. So from this olfactory bulb, it can give or send impulse directly to the brain. So the receptive end of the cells contains cilia. This one what I have said to you. The cilia of the receptor cells that extend to the mucous layer. So the movement of the transmission of impulse is from here, from the mucous layer, going upper to the part of the brain. Then we go to the olfaction. How it is transmission? It's from the chemical, what we call as the odorant. Odorant will diffuse and bind to the protein called as odorant receptor at the plasma membrane of the cilia. Okay. So at the cilia, we do have the receptor cells. So the odorant will bind here. The odorant will bind here at this part. And then it will depolarize the membrane and the receptor cells will generate action potential and further trying to transmitting the impulse towards the brain. So second one, we do have gustation. Gustation is we do have the gustation receptor cells. Where is it? Okay, so it is in the taste bud. Okay, so at the tongue, we do have all the buds there. Okay, it's like one dot dot. You can see there are two cells of taste bud. First one, taste cells, or what we call as gustatory cells. And second one, it is basal cells. Okay, this one is the basal cells. Okay, at the tongue. Okay, if we try to enlarge the papilla itself, okay, inside the papilla, we can see the basal cells. Okay, the one is in a green color, and then we do have taste cells. The taste cells, or what we call as the gustatory cells, are the cells that projects the hair from the tips of the taste cells to the taste. Okay, dia akan projection there. The projection will be there. So the basal cells are like of stem cells. Stem cells mean the one that can differentiate further to divide, differentiate into gustatory cells or into the taste cells. Means after it matured enough, it can become a taste cells. Okay. So there are 5 taste sensations from sweet, sour, salty, bitter, and umami. So where is it? At sweet, we can at the tip of the tongue, sour at the side, the salty at the side, but in a part of it. At the back of the tongue, we can feel the taste of bitter, and nearly to the pharynx, there's umami stick. So the substances can cause can bind to the protein receptor, and the detection can triggers the signal transduction pathway, whether it can depolarize the cells, how the influx can cause the membrane permeability and depolarize the cells. Mostly all of those mechanisms is the same. For the first, how it is triggered. Second, the binding to the receptor, then depolarization occur, then the transmission of the impulse occurs. Okay. Then we go next to the third part for the eyes and vision. So the eyes, for the structure of the eyes, you do have 3 layers. Okay. The structure of eyes is spherical in shape. It's not circular, but spherical, almost circle, but it's sphere in shape. They are fluid field structure, means all of those inside there, inside this inside this space is what we call as fluid. Mostly here, also fluid there. And it is enclosed 3 layers from outermost layer, middle layer, and innermost layer. Outermost layer, you see there, the one that I colored in green are sclera and cornea. We do have this sclera, the one that I colored in green. So this one is the sclera and cornea. So cornea itself here. Then we do have the middle layer. Middle layer is choroid. Okay, the one is see in black in color, choroid. If it is in front, it is ciliary body. The one that keep the iris, the pupil will open, dilate, or constrict based on this ciliary body pigment. Then we do have the third or innermost layer of retina. Retina is in red in color there. So the retina is rich in nerves and so on. So there are 3 layers of eye. So go for the first part, sclera, the tough layer of connective tissue. It can visible, okay, the white R's is from the sclera and forms visible part anteriorly, and outer part consists of transparent cornea. Okay, then we do have choroid iris ciliary body at the middle part, middle underneath sclera. They are highly pigmented and contain a lot of blood vessels that nourish the retina. And anteriorly means anteriorly it can form ciliary body and cornea. So this is the middle part. Retina is the outer pigmented layer and inner nervous system layer. Okay, means it's at the back, it contains a lot of nervous tissue. So here also that contain photoreceptor, means the receptors are sensitive to the light. Okay, it can contains whether it rods and cones. Okay, most of this one is bipolar neuron. Bipolar neuron. Okay, then we do have pupil and iris. Okay, pupil and iris. Okay, they are the one that can constrict and dilate. Constrict and dilate. The iris, the one that give you the eyes color, right? Whether it is blue or hazel and so on. So it's based on the color of the eyes, based on the iris. Iris are the visible ring structure that can increase or decrease in diameter. Whether it can constrict means can close, or dilate means it will open. So the constriction or opening is based on the contraction of muscles. Okay, both of them because of the contraction of muscles, but different muscle. Okay, based on different muscle. For the constriction is because of the circular muscles via parasympathetic stimulation, but if it is dilation, it's from contraction of radial muscles of sympathetic stimulation. Then we go to the lens. The lens is like the contact lens. Once we want to try to adjust the your sight and so on, so you do have the lens, right? So the lens there are suspended from suspensory ligaments and attached to the muscles called the ciliary body. Okay, the ciliary body there, the ones that directly related to the sclera part. Okay, so the lens. Okay, we do have the ciliary body that control the lens. So the lens here. Then we go to the interior of the eyes. Okay. There are 2 fluid cavities separated by the lens. We do have cavity, vitreous humor. Okay, this one is the first cavity. Okay. Second one, we do have aqueous humor. Okay, aqueous humor and vitreous. Okay, this one is the cavity that fill in the with what we call as the vitreous humor. Okay, so we go for the first part of what we call as aqueous humor, the one that I colored in red, aqueous humor. There. Okay, aqueous humor will fill the anterior chamber between cornea and lens. They are contained clear and watery fluid, and they are secreted by ciliary body there to provide nourishment to lens. Okay. So it can drain into canal of sclem, and if it is an adequate drainage, it can cause glaucoma. Then vitreous humor, the one that I colored in green there. Sorry, the one that I colored in green there. Vitreous humor. Vitreous humor is semi-fluid like jelly. It's like jelly structure, maintain the spherical shape of the eyeball and can transmit light. So if the light are coming from here, it can transmit at the back of the eyes where transmit into the retina part. Okay, retina. Retina do contains neuron axon and gathered point called at the optic disc as blind spot. Blind spot, lack of photoreceptor, blood vessel also can enter there. So we do have where it can focus and so on. So how when the light pass into the eyes, how's the transmission of the pulse output? Okay, so for the first part, once the light passes through the cornea, okay, this one cornea, and it go to the aqueous humor. Okay, for the first part, we do have the aqueous humor. Okay, the part that do contain fluid there. So it pass through the pupil. The pupil are the one that will open, whether open largely or try to constrict, because it can change how much the light can be. Whether if you are at the place that sun to has a direct sunlight, so you try to change the shape, whether you can allow more or less lighting. And it can pass through the lens. Okay, the lens are here. Now from the 1, 2, and 3, can change the shape to focus the image. This one where we can focus the image, whether it is blur or sharp image. Then it can travel through this, what we call as the vitreous humor, the jelly-like fluid. Finally, it can touch the retina where the photoreceptors are found. So, from cornea, aqueous humor. Okay, so try to answer which pathway for the first one, starting from cornea, aqueous humor, go to the pupil, to the lens, vitreous humor, and to the retina. These are the structure and composition of the eyes, and most of them do different part of location and give a different function of eyeball. Okay, then what are retina and where's the phototransduction occur? So now we want to focus more on the retina. Okay, retina where the blind spot are there. Okay, the retina, the one that at the inner part of the eyes. So the retina there can give phototransduction, means transmission of impulse when they got the light inside the eyes. So phototransduction means they want to convert from the light into electrical signals, means transmission of impulse. Okay. So the photopigment will undergo chemical reaction that activate by the light receptor potential in photoreceptor. From there, from the light, it can activate the receptor. So from this receptor, it will give potential to the photoreceptor. The receptor can generate action potential. Action potential there can transmit it into electrical signals. Electric. And from there, the ganglion cells will transmit info to the brain. There are layers of retina from the ganglion cells to the bipolar cells. Okay. So actually it starts from here, from the ciliary capillary to the pigment epithelium. We do have rod and cone. And then from here, from the light, it can transmit or change it into electrical signal. And the photoreceptor can contain rods and cones. They are sensitive, like the inner light sensitive ends face the choroid. And this synapse on the middle layer of bipolar cells, which synapse on the outer layer of the ganglion cells. Okay, actually, here we go. Okay, these are the layers of the retina itself. So the light will come from here, going to the ganglion cells. Then it is transmitted through the bipolar cells and detected by the photoreceptor. The photoreceptor here can generate action potential that transmit this light into electric. Then we do have to have two types of bipolar cells, what we call as rods and cones. So that after this we can see what are rod, what are cone. Then going to the pigment epithelium. Okay, rods and cone. Okay, for the first type, rod and cones outer segment full of latent discs with photopigmented molecules at the inner segment house of the cells to metabolic machinery. Okay. Okay. For the rods, it's like shape. It's like rod structure. One rod, right? Rod structure. But for the cone, it's like cone structure. It's what we call as rods and cones. Why we should have rods and cones? For the rods, it's allow the black and white vision in low light. For the cones, it's the receptor for bright light and provide high resolution for color vision. Okay. For rods, it's for the black and white. But for the rods, it's sensitive to the light but not provide sharp image for color vision. But for the cones, it can give for color and vision. And it contains pigment of rhodopsin for the rod. For the rod, it contains the rhodopsin pigment. Okay. And absorption causes rhodopsin to dissociate into retinaldehyde and opsin. So it's called as bleaching reaction. So this is the photoreceptor. Photoreceptor consists of rods and cones. You can see the outer pigment there. So you can see how's the transduction of impulse from the light. Okay, as what I have discussed before, the light will pass through the ganglion cells and then enter the bipolar cells before reaching the photoreceptors. Means this one side by side, 1, 2, 3. And then this area of photoreceptors are all area of the retina except fovea. Fovea is located at the exact center of the retina and cones only found here. Then we do have bipolar and ganglion cells layer that pull aside the light strikes the photoreceptor directly. Okay, you can see the photoreceptors are all in the area of retina. So go to the retina. Okay, retina. They contains all of the photoreceptor except here at the fovea. Fovea there do not contain photoreceptor. Why? Okay, why? Because only cones are found here. Okay, then they do have bipolar and ganglion cells layers at the fovea. Okay, so they do not have photoreceptor. This one is only for fovea that do contain this type, do not contain photoreceptor. So these are the direction of light from different illustration. Okay, from the light, it will pass through the ganglion cells and going to the bipolar cells. Okay, the one that do contain two extension from this neuron. So this one we call as the bipolar cells. And then it will transmit the impulse through the rod and cone. Okay. This is what we call as the photoreceptor cells. Here, the action potential will generate transmission of impulse from the light into electrical impulse. Okay, so this is the illustration from this. It's not from upper, but it's like you want to take it horizontally if the direction of light from here. So it's the same from the ganglion cells towards the bipolar cells, towards the photoreceptor cells, and direct back to the retina. So go for the adaptation of the light. When persons enter the dark room, so there are fewer photopigments in the rods and cones. But after 20 minutes, more visual pigments are produced. That's why they are adapting to the light. Then we do have light reflection. Okay, for the light reflection, it will transduce light energy into nerve impulse and only limited part of electromagnetic spectrum. Light rays will be diverged in all directions from very point of the light source. From these lights, it will diverge. Diverge means try to catch it and try to diverge in all directions from every point of the light point. The divergent light rays reaching the eyes must be bent inward. So it will bend inward to focus only at one part of the retina. So direction of light changes if it strikes the sphere surface, a new medium at any angle other than perpendicular, means you can see why it's larger in size if it is at the new medium. Okay, this is what we call as the light refraction. When the light passes from one medium to another, it will bend. So curvature of the point of refraction can also bend the light. So here we can see the curvature. Changing of the curvature of the lens will allow fine control of the focus. Okay, so this one is the one that are happening outside of our body. So it is the same principle there. We do have the light reflection because of the lens. The lens can control whether it can focus the image or not. So the image is flipped upside down in this process. So if you can see, it is here. So after the light reflection occur, the focus here will become upside down, like that. So image flip upside down during the process. So we can see the light refraction, if it is taller than, it will become at the focus point at the fovea centralis, it will flip down and it will become smaller because of the refraction that are occurring through the lens. Okay, the different light refraction of lens, whether it is convex or concave or convex. We go to the convex. Okay, convex is curves upward, upwards, and it will converge the light rays. Okay, from this direction of the light, it can converge, means it can focus at only one point. Okay, yeah, because of the convex. If it is concave, the light here, it is curves inwards and it can diverge all the light rays. So based on the physiology of the lens, it's require adaptation and challenges called accommodation, what we call as the ability to the lens to keep the object focused on the retina. So this process can increase the refractory power of the lens. So accommodation means the ability of the eye to change its focus from distant to near, or from near to distant by changing the lens shape. So here, if it is distant light source, if it's this one what we call as the lens that are convex in surface, it can focus there, the focus point. If it is near, so it can having a diverging rays, can try to alihkan these focal points not at the focus point. It will be deviated from it. It's stronger lens needed to focus on, means it can has more outward bend. Means the lens should be stronger, so the focal point can be return back to the normal stage of the focus. Okay, we do have physiology of the lens. Okay, focusing on the near object. If we are focused in the near object, for example, we want to try to read at the very near objects. So what are happening there? For example, okay, the ciliary muscles will be contracted, and then choroid will be broadly pulled towards the lens. Suspensory ligaments relax, lens will be more thicker. You can see here, the lens will be more thicker and also rounder because they want to focus at the only one focus point. Okay, hanya ada di sini saja. Ini yang betul. Kalau tak, kalau dia terlalu jauh, kalau dia tak baiki benda ni, focus point lari dan dia tak nampak benda tu. So macam mana dia nak baiki? Dia try besarkan dan juga gemukkan dia punya lens tu, maksud dia. It's become more curvature and more binding to the light and what we call small convergence of eyeball. If you want to focus on distance object, ciliary muscle, relax, pulls on the sensory neuron, ligament, suspensory ligament, and it causes the lens to thin and not so curvature. This is good for distance, means it's become thinner. So it is good for distance because they want to retain at the same focus. Okay. There are a lot of homostatic imbalance that occur. Refractive problems, myopia, or what we call as nearsightedness, and the distance image can be point in the front of retina. So the focus point is not on the retina because of this myopia, and image of blurry. Rabun jauh lah kata. And then we do have this myopia, the near image pula dia jadi into focus on retina without accommodation, and they are having near vision is better than the far vision. That's why we call as rabun jauh. It's because of the elongated eyeballs and it can be corrected by concave lenses. Concave, okay? Concave like this. Okay, this one. The surface should be concave. Means dia kena pakai spek mata yang concave in surface. So, it will become more divergence. Then we do have second one, hyperopia, because of the short eyeball. Tadi myopia because of the long eyeball. Okay, because of the long eyeball. But for the hyperopia, because of the long eyeball, short eyeball. What we call as farsightedness. Image are focused behind the retina means after the retina, it's the image are focused here. So means they have far vision is better than near vision. So this one is occur to the elderly, means orang-orang tua lah yang berlaku kat sini. Because of the short eyeball. And it can be corrected by the convex lenses. Then we do have astigmatism, irregular shape of the cornea. So the cornea or the lens itself is having a irregular shape. And how we want to correct it's by the cylindrical lenses. Okay. So this is all those myopia. So how we want to correct it? Which one is it, concave or convex? So for astigmatism, it's cylindrical. For hyperopia, it should have a convex lens. For the myopia, it should have or should be corrected by the concave lens. Ametropia is the one who are having normal vision and the ray are focused on the retina, so it is a healthy individual. Okay, for the hyperopia and myopia, okay, hyperopia, if the focus behind the retina. For myopia, it's focused at the in front of retina. So you should correct it by using for hyperopia. It should have convex lens, and this one should be corrected by the concave lens. Right. So now it can now be corrected. This is the correction, so that the focus point on the retina, not behind or directly in front of the retina. Okay, so thus I think it is for the first part of the eye and the side. So we continue this lecture on part 2. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.